Hey guys, my name's Alyssa, and I'm excited to share with you about the project I worked on this past summer, studying what factors might affect a cancer cell's susceptibility to irreversible electroporation, basically just electrical shock. So, irreversible electroporation, or IRE, in a clinic is performed by inserting two electrical probes on either side of a solid tumor and then conducting high voltage electrical pulses between those two metal plates, and then that delivers electrical shock essentially to the solid tumor. And at the cellular level, what happens is that the high voltage pulses disrupt the membrane potential of the cell membrane, causing pores to form in the cell membrane. And then this allows everything within the cell to be released into the surrounding environment, and that will, of course, cause the cells to undergo apoptosis or programmed cell death. And then once the cells have initiated this self-destruction process, the patient's immune cells will come in and clean up the cellular debris. We can model this treatment in the lab using cell culture techniques. And so the way I did this is by growing cells in a plastic flask. They attach to the bottom of the plate and grow in a media that has nutrients like amino acids and sugar, everything that a cell needs to grow. And in order to work with the cells, I can detach them from the plate and get them into a liquid suspension. And then I place them into electrical cuvettes that model electrical probes for IRE because they have two metal plates on either side. And so the sample of cells can be placed in an electroporator machine, and pulses of desired strength can be conducted between these two metal plates, and the cells can receive that dosage of treatment. And this models induction of pores within the cell membrane. And then the way that I can measure cell viability is by taking a small sample of the cells that were treated with the shock and mixing them with a dye called trypan blue. And under a microscope, cells that have pores in the membrane are more permeable to the dye, and so under a microscope, they will appear blue, versus cells that are alive and well have intact cell membranes and they exclude the dye, so under a microscope they would appear to be a cream color. And then you can compare the percentage of live versus dead cells in each of your treatment groups. So the first thing I did in the lab was I looked at two specific cell types. I looked at HeLa cervical cancer cells and A375 melanoma cells. And I just did a dosage curve of electrical voltage treatment. And so with HeLa and melanoma cells, as the applied electrical voltage increases, the cell viability decreases. But interestingly, to get a 60% viability where we're killing about 40% of the cells, HeLa cells required about a 300 volt treatment versus melanoma cells required a 500 volt treatment. And this just demonstrates that different cell types are more resistant than others. So what factors might affect a cancer cell's susceptibility to this treatment? We looked into chemical factors, which could be modifications to the cell's membrane, or physical factors, which would include previous exposure to the treatment, which is a major issue for all cancer treatments, is that cells can develop resistance over time. So it's important to study the mechanisms of why. So our first question was, do cancer cells develop resistance to this treatment after repeated exposure? And the way that we did this in the lab is, again, starting from the same cell culture population of cells, we got them into a liquid suspension and then split them into three treatment groups. One group would receive 3 shocks, and in between shocks, they would be plated into a plastic cell culture plate and allowed to recover with fresh media, fresh nutrients, and then they would be removed from the plate and shocked again. And then we had a population of cells that were shocked either 1 treatment round, 2 treatment rounds, or 3 treatment rounds. And what we would hypothesize is that the cells that had only been shocked with 1 round of treatment would have a lower viability compared to cells that had been shocked previously or had previous exposure to the treatment if the cells developed adaptive resistance. But what we found was that neither HeLa nor melanoma cells developed resistance to repeated treatment. But interestingly, the melanoma cells were a bit more resistant to 1 and 2 rounds of treatment, and we didn't see a significant decrease in viability until the 3rd round. Versus in HeLa cells, we saw a significant difference in viability in cells that received 2 rounds of treatment compared to cells that had only 1 round of treatment. And this also is indicative that not all cancer cell types respond the same to the treatment. Our next question was if chemical factors might affect a cancer cell susceptibility. So we altered this chemical environment by treating the cells with a statin drug, atorvastatin, that decreases total cholesterol in the cells. Normally, cholesterol is a molecule in the cell membrane that allows the membrane to be more structured or more rigid. The relative amount of cholesterol determines the rigidity or fluidity of the membrane. So we hypothesized that decreasing the total cholesterol in the cells through statin treatment would create a more fluid cell membrane, and this would potentially allow the cell to recover after electroporation insult to the membrane. So when the pores form, if the membrane is more fluid, it could potentially more easily reseal those pores and it could survive that round of treatment. And interestingly, with HeLa cells, we found that pretreatment with the statin drug did, in this population of cells, confer a higher viability after treatment with IRE compared to cells that were only treated with the solvent for the statin drug as a control. And so it'd be interesting to look into the mechanism of this, whether cholesterol in the membrane is actually decreased. And another notable point is that the paper we based our methodology on, they worked with pancreatic cancer cells, and they found that these cells had a significantly higher viability with repeated treatment. And that's the exact opposite of the results that we found, that with more treatment, the cells just had a lower viability. So this suggests that different cancer cell types have variable responses to the treatment. Another important consideration is that statin drugs are very commonly prescribed medications. They're one of the most commonly prescribed in the US. So it could be worthwhile to assess its potential effects on patients who are also receiving IRE treatment for their tumors. Their tumors may be somewhat more resistant to this treatment because of their decreased total cholesterol. In future directions, like I said on our previous slide, we would look for a more direct measure of cholesterol levels in the cell membrane, potentially with a microscopic assay that would allow us to see cholesterol through a fluorometric assay. And we could also look more specifically at the cell death pathway, apoptosis. We could look for expression of particular proteins that are involved in apoptosis to determine the mechanism of cell death that is involved. And I'd just like to thank the Lee McNair program for funding this project over the summer, Dr. Cornett for his mentorship and for being my advisor on this project, and to the Lee Department of Natural Sciences for the lab space over the summer, the socially distanced lab space. So thanks so much for listening.